Okay, so this is the second part to section 3 because I didn't want to make too long of a lecture recording. So let's talk about refrigeration and freezing. So cold doesn't kill most microbes. All it does is slow it down. So refrigeration, which is usually about 0 to 7 degrees Celsius, it stops the growth of most pathogens, but some, like Listeria, which we talked about before, and also Yersinia, they're perfectly happy in your fridge. If we're talking about freezing, freezing can be more damaging because ice crystals can actually puncture cell membranes, especially if we're talking about slowly freezing something. But many microbes survive frozen storage, so this is actually a good thing when we're talking about lab cultures, but not a good thing if we're talking about food safety. Okay, desiccation, we're talking about, again, removing the water or dehydrating something essentially, because this is really important because water is essential to all life, including microbes. So if we take water away, metabolism stops. So that's why drying has been used to preserve foods for like ever. So desiccation works for things like beef jerky or dried fruit, something like that. We also have lyophilization. This is essentially freeze drying, so freezing microbes and removing water via a process called sublimation. So it prevents those ice crystals that I talked about with freezing, and it keeps microbes viable for years. So it's why labs can keep strains on the shelf for ages without even needing a freezer. All they have to do is freeze dry it. So filtration is really important because sometimes the best way to control microbes is to actually physically remove them. So membrane filtration is really perfect when we're talking about heat sensitive fluids, so things like antibiotics or those containing vitamins or our culture media. So if we're talking about air, these are membrane filter sizes. I could care less about knowing anything from this table at all. If we're talking about air, usually we're going to be using what's called HEPA filters, so HEPA. We actually had them in our LTP building fairly recently. We put them in, and these are what HEPA stands for is high efficiency particulate air filters. And these are really important because they can remove 99.97% of the particles in the air. So not only does our labs and our building have them, but they're also kept in operating rooms, and they're often also used, like in this picture, in those biosafety cabinets that we talked about in our BSL levels. Okay, so osmotic pressure. So a lot of times food is packaged with salt or sugar, and the purpose of doing this is to create a hypertonic environment that draws water out of cells. So again, if we draw water out of cells, we are stopping metabolism. So that's why salted meats, honey, and jams, why they last so long. Bacteria can't handle osmotic stress, but molds can. So sometimes you can see mold buildup on your jelly that you forgot about in the back of your fridge, like I just discovered last week. Okay, radiation, it's a really powerful microbial control tool. We've talked about some of these. We can have ionizing radiation, so things like gamma rays, x-rays, electron beams. They all cause electrons to be ejected. So essentially, they cause irreversible damage to DNA and proteins. So it is a really great tool for sterilizing medical supplies and some foods. We also have non-ionizing, what we call UVC light, and that doesn't penetrate as deeply, but it does create those pyrimidine dimers in DNA and blocks replication. So again, we talked about these previously, and those are really good for disinfecting surfaces. They can also be used to disinfect just the air and disinfect water. Oh, there they are there. So this table is just kind of giving you a overview of the methods that we just talked about. Like I said, you don't need to know any specific temperatures, just a general idea of what all these terms are.